Да, 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 давайте. То есть я потом просто выключаю все и все, да? Хорошо, большое вам спасибо. То есть вам не надо больше входить сюда. Хорошо, Дима, большущее вам спасибо. Я начинаю, да? Спасибо большое. До свидания. Guys, good morning or good afternoon. And today you are going to see the lecture on the gram-positive cocci. That is a really very large group of bacteria. And they are not all equally of medical importance, but the most important are, as you see here, Staphylococci and Streptococci. So, and we usually discuss them together because quite often from the very beginning when we are going to diagnose the disease, we have a problem with differential diagnosis between Staphylococcus and Streptococcus because they may usually cause mixed infections or similar infections. So from the very beginning, we need to know whether we direct our identification towards Streptococci or Staphylococci. And from the very beginning, we could see that they have distinct morphological features, mainly regarding the arrangement of the bacteria in a smear. And you might remember already that this depends very much on the way how bacteria divide. If they divide in one plane, like diplococci or streptococci, or in two planes like tetrads, micrococcus to differentiate with the others, or in three planes like staphylococcus, and the aggregation is very typical for staphylococcus grape-like clusters. So resistant Staphylococcus aureus should be completely isolated within the hospitals because it can be easily transmitted, the Staphylococcus, and it is really very difficult to get rid of in hospitals and wherever else, right? Okay, and of course, out of all gram-positive cocci, we will pay more attention at Staphylococcus. Cocci quite heterogeneous, and this group consists of more than 16 genera, and cocci have common features. Of course, they are spherical in shape and they are quite small. Then they are gram-positive after gram staining, and they cannot form endospores. And we won't care so much within our lecture about the other genera, but we concentrate on Staphylococcus and Streptococcus. And from the very beginning again, I would like to underline that we would like to, on every step of our um, lecture or discussion, we would like to see the differences on every single stage in between Staphylococcus and Streptococcus. So the first one is how can we differentiate the genera on the level of the genus, okay? So, and in this, except for arrangement under the microscope in a smear, we may base our presumptive differentiation into the ability to produce enzyme catalase. And you can see that Staphylococcus is catalase positive and Streptococcus is catalase negative. Catalase, the enzyme that split the hydrogen peroxide, right? So, and if we place a drop of hydrogen peroxide on the slide and add a loopful of the colony of Staphylococcus, the reaction with the release of oxygen enable to split hydrogen peroxide. So that is very simple and very informative test to differentiate on the uh, level of genera in between Staphylococcus and Streptococcus. Now, step by step, we discuss them and we start with Staphylococcus. And you can see it here in the smear taken from the patient. Why it is so? Because you see the elements that cannot be seen in pure culture. These are the pus cells, right? Because usually Staphylococcus causes pus or purulent infections. And you can see very well the bunch of grapes, and this is type of arrangement, and the name Staphylococcus refers to the fact that the cells of this gram-positive cocci arranged in a cluster resembles a cluster of grapes due to the cell division in three planes, right? And one more feature that the daughter cells stay tightly attached, stay in the close proximity because they have a special factors on the surface that uh, lead to aggregation of the daughter cells. So, and from the very beginning, Staphylococcus, I mean, if you are talking about Staphylococcus species, there's a large collection of many species, right? So, but then they can be broadly classified into coagulase positive, and there we have the one that it could be highly pathogenic, Staphylococcus aureus. And we have also coagulase negative. And here you can see a lot of species that even from their names, species names, you may guess that they are not so highly pathogenic or not pathogenic. Staphylococcus epidermidis, saprophyticus, and xylosis, and the others, right? So, but then the test that helped to distinguish potentially pathogenic and non-pathogenic staphylococci is the coagulase test. If we have taken citrated plasma, citrate is added to prevent the natural coagulation of the blood serum, and we inoculate it with the bacteria and incubate overnight. Then, if it is Staphylococcus aureus, it releases coagulase enzyme that turns plasma to clot or blood to clot. And if there's no coagulase is produced, so the citrated plasma stay as a liquid, and the coagulase turn it into a gel. So the Staphylococcus aureus can cause a wide variety of the infections, and to classify them somehow, they can be broadly subdivided into three major types. First type are the superficial lesions such as wounds, infections, and they are really the Staphylococcus aureus is very common cause of these um, uh, soft tissues infections. Then we distinguish a group of so-called toxinoses, the conditions that are usually formed not by just multiplication of bacteria, but the ability of the staphylococci to release exotoxins. And the exotoxins have different mechanism of action, so and those diseases seem not to be related because they are due to the particular type of toxin. And the examples are food poisoning and some other health condition that we discuss later on. And the third group uh, is for most dangerous, systemic, and life threatening conditions, such as endocarditis, osteomyelitis, pneumonia, brain abscesses, meningitis, and bacteremia. But by the way, this is maybe a result of superficial infections initially. So now, what kind of diseases are caused by staphylococci? So we start with the first group, skin and soft tissues infections, right? And the Staphylococcus is very frequent cause of the folliculitis, furunculus, carbunculus, wound infections as a result of trauma or surgical operation, and impetigo. And just take a look at this, how this infection appears, right? So you can see the most common one, folliculitis. So that refer to as pimples on pustules, where the Staphylococcus aureus infect the hair follicles, right? And that is really common. Then a sty, a pus-forming infection of a gland of the eyelid that is often caused by staphylococcus. Then we go ahead with the exterior abscess that is called a furuncle. This is a large, deep pus-filled infection, and you can see that distinctly. We can see encapsulation of the abscess. And by the way, the encapsulation of the organism is really very typical for staphylococcal infections because the tendency to aggregate, right? So, and this encapsulation usually prevents spreading the microbes into the blood. So on one side it is a good news, right? So that the infection is encapsulated, but on another side it is not reach these bacteria once they are encapsulated by the circulating antibiotics if given systemically, right, from the blood. So then this is rather surgical disease because the abscess should it is necessary to lance and drain abscess surgically and then apply antimicrobial drug. Then it works, but not like this. So then we can see carbunculus, an infection of cutaneous and subcutaneous tissues that consist of cluster of boils, often caused by staphylococcal organisms. And also the surface infection, superficial vesicular-pustular infection of the skin, is an impetigo. And it is found very often on the arms, legs, face, and this is high contagious pyoderma. And by the way, it is very commonly appear as mixed infection with Staphylococci and Streptococcus, either of them or both together. And in the beginning, just redness of the skin, but it progress rapidly to the itching blisters. We can see the breakthrough and crusts on this. So these were the soft tissues infection. And now we take a look at the toxin mediated infections. And at least these three we have to consider. This is called skin syndrome, toxic shock syndrome, and food poisoning. Okay, and then we have a third group of the diseases that is systemic and life-threatening cerebral, CNS infection of the central nervous system, and that's usually as a result of bacteremia, because bacteria can be spread inside the macrophages through the BBB. And of course, there's a very common pulmonary infections as pneumonia and paima of the pleura, and that is very common complication of, let's say, influenza virus infections, where as a result of primary infection, staphylococci get access to the not just upper but also lower respiratory tract. So the Staphylococcus aureus is really famous for its mucoskeletal infections. It is a very common cause of osteomyelitis. So gut infections are also genitourinary tract infections are usually very common, and it may cause cystitis, but it also may cause viremia when bacteria of staphylococci. So it can grow on ordinary medium, but the growth will be very scanty. So that's why usually for uh, culturing in the laboratory, we would like to use enriched media, enriched with blood, serum, sugar, something like this. So the temperature is usually regular 37 degrees Celsius, and the uh, medium should be neutral or slightly alkaline. These bacteria are facultative anaerobic bacteria, so they may grow with or without oxygen. And the name Aurus, Staphylococcus aureus, means they may produce the golden yellow pigment that is called staphyloxanthin. By the way, this is carotenoid pigment, but it may act as a virulence factor, primarily by as an antioxidant that help the microbe to stand uh, mm, the active radical that can be found inside the macrophage vesicles, lysosomes, so to kill bacteria. So it may stand it due to this cephaloxanthin. So quite often, because staphylococci um, could be isolated from contaminated specimen, like oropharyngeal swabs, for example, we need to use selective media. And these are yolk salt agar, salt milk agar, or tellurite agar. So in tellurite agar, uh, Staphylococcus stand the salts very well. So and it may reduce telluride, producing black colonies, right? And we also often use blood agar, but this is as enriched media and also as differential media to demonstrate ability to produce hemolysis. So the Staphylococcus aureus is a good model to study biochemical reactions that can be used in identification of these bacteria. So we mentioned coagulase already, and stay again, you can see coagulase positive and coagulase negative reaction. Then Staphylococcus aureus may ferment sugars, including mannitol, not only aerobically, but also anaerobically. And that is, by the way, an important feature, because the only highly pathogenic staphylococcus can ferment mannitol not only in aerobic, but also in anaerobic conditions. So it is specially performed to confirm the pathogenicity of the microbe. Then um, Staphylococcus aureus may produce enzyme that is called lecithinase, and we demonstrate the ability to produce it on the yolk salt agar. And lecithinase enzyme will modify lecithin that is one of the constitution of yolk salt agar. And then you can see the opaque halo around bright yellow colonies, or you can see the opaque halo along the streaks or around the streaks or colonies of Staphylococcus. Normally medium is transparent yolk salt agar, but then when lecithinase modify lecithin and turns it into the non-soluble precipitate, so you can see this precipitate as an opaque halo. So it also may produce some thermostable nucleases that may cut DNA, and they also may liquefy gelatin. So these are uh, not just peptolytic but proteolytic properties, and also produce some others. So we see some reactions. So this is a mannitol fermentation. So the test distinguishes pathogenic and non-pathogenic staphylococci. So, and as I said, the sign of the mannitol fermentation is the changes over the pH indicator, where the medium turns uh, acidic, and depending on the type of the pH indicator and the medium we use, it turns, for example, from red to yellow, then it will be a sign of positive reaction. Now, we mentioned some factors that can be considered as pathogenic, but now let's take a closer look at the pathogenic factors that are seen in Staphylococcus aureus. And here you can see a simplified scheme of the genome over the Staphylococcus, and you can see here in this genome the basic genetic element, that is a chromosome, and many extra chromosomal genetic elements incorporated into the chromosome. And you can see, so the bacteriophage, you can see transposons and phage and phage here, and you can see the plasmids, and also you can see some free plasmids that also may occur inside the mm, mm, staphylococcal cell. And by the way, when we studied genetic, we stated that the uh, the majority of the virulence factors are encoded by um, extra chromosomal genetic elements like transposons, plasmids, and bacteriophages. So we can see it here that some cytotoxic factors, these are the factors that will be secreted to outside the cell, the bacterial cell, right? The Staphylococcus aureus are the production of these factors is regulated by the plasmid DNA, or by it is encoded by transposons on bacteriophages. Among those factors governed by the extra chromosomal genetic elements could be the leukocidin, the enzyme that cleaves the or causes death of the leukocytes, many proteases, enterotoxin that may cause food poisoning, exfoliative toxin, and toxic shock syndrome toxin that cause toxin-mediated diseases. So they are from these genetic elements. Then, of course, antibiotic resistance is tightly connected with the, for example, plasmid that may cause resistance to methicillin, and also the plasmids that govern the production of beta-lactamase against beta-lactam drugs and other uh, antibiotics. So, then we also may have the surface factors. So these are secretory factors. We also may have the surface factors. They are connected with the ability to produce adhesins like fibrinonectin binding adhesins, then fibrinogen binding proteins, and also some other factors that may mediate adhesion. And here you can see the list of basic diseases that are mediated by these virulence factors. So now, few words about the surface factors. If you take a look at the simplified scheme of the staphylococcal cell, you can see that on the surface of the cell, we may see some factors that may help in on different stages of the staphylococcal of staphylococci to cause diseases. Again, we mentioned adhesins that are cell-bound proteins. Also, the important adhesins are teichoic acids that, of course, governed by the chromosome because it is a part of the cell wall. Then adhesins connected with binding with fibrin, fibrinonectin, and some of them may cause tissue damage. And some microcapsule that can be attributed to some Staphylococcus aureus species. So, and the some proteins in the cell wall. For example, you can see here the so-called protein A that is incorporated into the cell wall. So, the very important feature of the protein A that it has very high affinity to Fc portion of immunoglobulin G. Do you remember, my dears, you have studied in the biochemistry? So, and these are FAB portions by which they usually bind with microbe, and FC portion that helps macrophages to find this complex and to engulf it. So, but then the protein A has affinity to FC portion, and uh, the cell of Staphylococcus aureus decorated in this way by immunoglobulins G will escape the recognition by other immunoglobulins, and so it may escape the labeling with antibodies and phagocytosis. Now let's take a look at some groups of the virulence factors. So we mentioned adhesins. These are surface macromolecules that promote adhesion and colonization. And we can see here some of them. The capsule or the capsule material is not really very heavy, but it has high adhesive properties to synthetic material and to the human cells also. And it is believed that the capsule material of Staphylococcus aureus may inhibit the activity of macrophages in diapedesis and may inhibit the activity of killing microbes inside. Then we have a thick layer of the peptidoglycan, and it is gram-positive bacteria. It means it doesn't have endotoxin, but it is believed that peptidoglycan of Staphylococcus aureus may have the activity that resembles the activity of lipopolysaccharide or endotoxin of that of gram-negative bacteria. So it may activate complement and activate macrophages to produce interleukin 1, so it may cause very strong inflammatory response in the body. Then we can see teichoic and lipoteichoic acids that bind to fibrinonectin and acts as very strong adhesins. They also may participate in activation of the complement system and also may perform so-called clotting of kallikrein kinin system in the blood. Then we mentioned protein already. Here it is shown as black one, right? And this is the IgG that decorate it and prevent recognition of the specific recognition of the staphylococci. Then also I would like to mention bound coagulase, or so-called clumping factor. Here it is shown purple. So this is the factor that binds fibrinogen and causes Staphylococcus to clump. That is one of the reasons why it segregates and start to be encapsulated in the human body. So then you can see that those adhesins and structural components of Staphylococcus aureus cell wall mediate a so-called biofilm formation of the indwelling catheters, for example, and this is really very dangerous for intravenous and urinary catheters, right? So, but also may make this biofilm within the human tissues. Then we go ahead with the virulence factor, and straight after the adhesion and colonization, these bacteria may cause invasion of the deeper tissues. So we may consider the factors of invasiveness or so-called invasins that promote bacterial spread in tissues. And here we have a lot of toxins and enzymes. The examples are hyaluronidase. So now we will take a look at them. No, not now. Okay, later on a bit. So then, the next group of virulence factor are aggressive factors or factors of aggressiveness. So these are the factors that allow bacteria to survive within the human body and evade the innate and acquired immunity mechanisms. These factors may inhibit the activity of phagocytes, the migration of phagocytes toward the place of site of inflammation. Then they may also inhibit the ability to engulf bacteria and so on. Then some other aggressive factors may help uh, to survive these phagocytes inside the uh, macrophages. For example, staphyloxanthin may inhibit uh, the active radicals that may damage the cell wall over the Staphylococcus aureus and may lead to digestion of Staphylococcus. So staphyloxanthin and catalase are examples of it. Now we take a look at the staphylococcal toxins, cytotoxins. These are very important virulence factor, mainly attributed to soft tissues and muscles infections. So we have a group of cytotoxins, and they all have a similar mechanism of action that is called membrane damaging toxins. It's really transmitted or engulfed by the cell. No, they act upon the cell membrane, not entering inside this human cell. But then if membrane is damaged, so then of course the cell gonna die of osmotic lysis. So out of those, I would like to mention these, they will be responsible to tissue damage. Then we have beta toxin, lipids, and catalyzes the hydrolysis of phospholipids. And again, it is responsible for tissue damage and abscesses formation. So delta hemolysin or delta toxin and gamma toxin. So they have wide spectrum of cytolytic activity. They may act in the way similar how the detergents work. And they may lose lymphoblastic cells, human, sheep erythrocytes, and they are also responsible for skin and soft tissues infections. And finally, we have leukocidinoga, beta toxin that can cause lysis of RBCs also, but it is a multi-component protein that produced as separate component and separate components, and then when come together, they act against the human cell membrane and increase permeability uh, in mainly leukocyte cell membranes and cause the cells to die. Now, a group of separate toxins with the particular mechanisms of action. The first one is so-called exfoliative toxin, A and B. And it may cause local and disseminated manifestation. This is disseminated manifestation, and the health condition is called Staphylococcus scalded skin syndrome. So this is a disease characterized by exfoliative dermatitis. So how toxin works? You can see the normal skin structure, and you can see that the desmosomes connect the cells here, and if the SSS toxin is released, the toxin causes the damage to the intracellular bridges or desmosomes, see here, and uh, due to this we have exfoliation of the upper part of the stratum uh, uh, epithelium. The toxin activity doesn't cause the lysis of the cells, but then uh, when it occurs in the systemic way, so the release of this, the exfoliation of the upper part of the skin may lead to secondary infection. Then the toxin doesn't really cause very strong inflammatory response, but recolonization of the skin of secondary pathogen may lead to very serious conditions. And here you can see the localized form of the scalded skin syndrome toxin, and this is called bulo impetigo. And usually it is produced in children not older than 5 years. The mortality rate is really very low unless the recolonization of the damaged skin with secondary pathogens may happen. Then it may lead to death. Here you can see the result of the activity, and this is post-mortem picture of the uh, staphylococcal toxic shock syndrome toxin. And this uh, staphylococci may infect the hyperabsorbent tampons, nasal and vaginal tampons, and while these strains of Staphylococcus aureus multiply there, they produce this toxin that may cause very severe health conditions, starting with fever and hypotension, rash, then discoloration of the skin, and multi organ failure. Then we may have renal, hepatic failure, and so on. So apart from the other activities, the toxin may act as super antigen. We haven't studied immunology yet, so that's why maybe we need some explanation of it. What normally happens when the antigen presenting cell, like macrophage, recognize specifically the protein, process it, and present this antigen to T lymphocytes. Then we have an activation of both macrophages and T lymphocytes in a highly specific way due to the recognition of particular antigen. So very few T cells will be activated by these macrophages, right? But then it appears that super antigens, like is shown here, may bind from outside the immunological synapse. non-specifically to the viable regions over the uh, MHC2 and T cell receptor. So, and by this, T cells will be stimulated and macrophages will be stimulated to produce very important uh, interleukins. So, for example, macrophage will produce interleukin 1 and tumor necrosis factor that may lead to very strong inflammatory response. But then super antigens may lead to the activation of not just 1 out of 5 T cells if it happens um, not in one in 10,000, how it happens in specific activation. But 1 out of 5 T cells will be activated by super antigens. So this amount of activated T cells and macrophages may lead to the uh, uh, dysregulation of the immune interleukins and cytokines production due to the development of the immune response and the release of large amount of pro-inflammatory signals. They may lead even to the very strong disbalance and the death of the patient. And this is like cytokine storm that we consider for coronavirus infection, by the way. Staphylococcal enterotoxin that cause food poisoning also can be considered as a super antigen. But it also an interesting toxin regarding the ability of this toxin to be stable for heating, to stand heating. And it may not be inactivated within 30 minutes at 100 degrees Celsius. It means if the food is prepared and stored not in proper conditions where staphylococci may multiply, even then if this food is preheated before serving the meal, still the toxin will be there. And this enterotoxin is a very strong inducer of interleukin 1, so that's why usually in staphylococcal food poisoning, so we have the very strong inflammatory response and very high temperature development. And it also stimulates the intestinal peristalsis and usually may cause CNS effect in the form of nausea, intensive vomiting. So staphylococcal toxins may be divided into 6 to 8 serological groups. What does it mean, serological groups? They may be divided according to their antigenic structure. So, and some of the enterotoxins are considered as toxic shock syndrome toxins and may have superantigen activity. So, few words about invasive enzymes that are common in staphylococci. So we mentioned already coagulase and catalase. So I would like to mention hyaluronidase. That is enzyme that hydrolyzes hyaluronic acid and facilitate the spreading in tissues. How this may work, so this is a simplified picture of the ability to produce particular enzymes. When we have bacteria inoculation into the soft tissue, they produce coagulase and cause plasma to clot around the staphylococci. They can multiply there, but then when they form this clot, so they are inaccessible by macrophages and antibodies. And then the kinases can be produced that may cause dissolving of fibrin clot, like staphylokinase, right? Or fibrinolysin, and that may help staphylococcus to be released into the bloodstream then and spread through the bloodstream. So one of the staphylococcal enzyme that important to develop very severe health conditions is penicillinase, by the way. We have studied it when we studied genetics, because this enzyme is called beta-lactamase also, that hydrolyzes penicillin, so it destroys the beta-lactam that is uh, the effective site of the penicillin, so and it is encoded and transmitted by the plasmids. Apart from this, a lot of lipases, nucleases, and proteases are produced by Staphylococcus. How do we perform laboratory diagnosis? We spent enough time to describe the different biochemical properties and enzymes, so you blame it as a causative agent of the disease. So for this, we need to identify the species name that this is Staphylococcus aureus. But apart from this, we may have a lot of non-pathogenic Staphylococcus aureus organisms that quite often can be a part of the normal flora. So they may lack very important determinants of pathogenicity, so they can be considered normal flora. But if they keep all those, especially extra chromosomal genetic elements that code for different and distinct virulence factors, then it will be highly pathogenic. So, and because all staphylococci are indistinguishable due to the morphological features, we have to perform usually culture for diagnosis. So, and the identification is made mainly based on the biochemical characteristics. From here, I would like to tell you that can be a big difference with the streptococcus, as usually we compare them, right? So, and as we've seen, the identification of staphylococci is based on biochemical activity, but identification of streptococci is based mainly on the antigenic structure. That is a difference. And the type of hemolysis. And only in few cases or in some research work, but usually not in clinic, we use biochemical characteristics. They are not that stable and important diagnosis like we keep them for staphylococci. Okay, and of course, while we identify the microbe as Staphylococcus aureus, pathogenic one, we have to perform antibiotic susceptibility tests. So, because it can easily develop resistance, so that's why we need to know with what particular drug we have to treat this particular patient. Phage typing is used for epidemiological needs to trace the source of infection. Do you remember? We have discussed it. So, if we have the hospital outbreak of staphylococcal infections, for example, we have to check the phage susceptibility of the strains isolated from the patients and maybe from medical personnel in the hospital. So to see the whether the phage they belong to same phagovars or phagotypes, or they belong to different phagovars. If to different, so they are not related. If they have the same susceptibility patterns, so then they are obtained from the same sources. So that's what we do when we need to trace a source of infection. So how our body try to protect or defend itself from the staphylococcal infection. That is important. How our body is going to kill microbes. What are the major effective ways for it? So generally, against Staphylococcus aureus and other conventional bacteria, phagocytosis is a very uh, important tool of protection. So, and also we consider this is from innate immunity mechanisms. And also antibodies are very important, especially for two reasons. First, antibodies against all those soluble protein factors may neutralize the toxins and dangerous enzymes. But then in another way, antibodies may opsonize or bind to bacteria, and by this they mark the bacteria to be recognized by better recognized by macrophages. So antibodies are very important. But then the problem is that staphylococci may be difficult to kill because of the carotenoids that we mentioned and catalase that make the killing not so effective, so they may survive in macrophages. So the treatment is usually performed by penicillins if they are effective, because generally they are very susceptible to penicillins. Unless the transmissible plasmid, they keep information about beta lactamase is there. It gives the resistant to staphylococci. And also, it is important that most of coagulase negative strains now that may circulate in the hospitals are resistant to newer penicillins like oxacillin, methicillin, and that also may occur due to the mutations in chromosomes that may convert the penicillin binding protein and they start to be non-recognizable. So usually if we have these multi-resistant strains of staphylococci, the drug of choice will be vancomycin. That is solved that the drug of choice for penicillin-resistant strains of Staphylococcus aureus. How else we may perform treatment except for antibacterial drugs? For this, we may perform sometimes so-called immunotherapy with neutralizing antitoxic antibodies. So the antibodies can be available for enterotoxin that may cause food poisoning, for toxic shock syndrome toxin, but also please try to distinguish it. We may have antibodies against the toxins, but we also may use so-called anti-staphylococcal immunoglobulins. So in some long-term chronic systemic infections, and these will be antibodies that bind with the surface of staphylococci themselves. It means this is antibodies directed against surface structures of the bacterial cell wall, of the staphylococcal cell wall. And we need to distinguish it from antitoxic antibodies. So usually hyperimmune serum or monoclonal antibodies against surface components may partially prevent the adhesion of staphylococci and may help in phagocytosis, but it is not very highly effective. So how can we prevent disease? By now, we don't have very good vaccines, protective vaccines available. So then we may perform immunization against toxins. That is possible, but then it is not widely used in the medical practice. Now, let's take a look at streptococci. So they are also gram-positive cocci from the same group, but they look differently under the microscope when we examine the smear of the pus, and you can see still some elements of the pus, and among them we can see uh, the streptococci arranged in pairs, short or long chains. And the name streptos is derived from the Greek term meaning twisted or coiled. You can see the twisted like chains or filaments. Right? Oh, here, twisted chains, right? So it's because the streptococcus divide in one plane when perform binary fission, and daughter cells fail to separate completely because still they have a lot of adhesive molecules on the surface. So they form these coiled or twisted chains. So we have a lot of streptococci. So that's why it is very difficult to, do we have? Okay. That's very difficult to classify them. So then from the very beginning, I would like to underline the basic way of streptococci classification is not biochemical properties. The basic way is serological classification, or so-called Landsfield grouping. And you can see here the name of some groups. Altogether, we have 17 groups from A to blah, K or something, right? So, and just some first, the names of the first groups is given. They are named by Latin letters, and they are grouped into A, B, C, and so on, zero groups by the precipitation test to reveal surface antigen. So it means the grouping or classification is made based on differences in antigenic structure. Then the second way that is common in clinic, Landsfield grouping, and the second way is studying the hemolytic patterns. So because all streptococci can be broadly classified into alpha hemolytic streptococci, beta hemolytic streptococci, and non-hemolytic. So then we demonstrate the type of hemolysis typical for this particular strain. And the answer or the result of the identification may be from, for example, may sound as group A beta hemolytic streptococcus without the species name. So then in some cases when we need a species name, then we perform biochemical identification using some biochemical properties that are not very stable in mainly streptococci. Okay? So 3 ways of identification. So when you study immunology, then we will take a closer look at the Lancet classification. Just now, I would like to say that some streptococci cannot be classified again by this classification. See, so Streptococcus pneumonia is not classified. Why? Because it has a very heavy capsule. So then it interferes with the finding of the cell wall antigen by precipitation test. So, or maybe it is mimicked or modified, so we cannot identify. So then, for this particular species of Streptococcus, we perform hemolytic pattern studying and biochemical identification of this Streptococcus. So, patterns of streptococcal hemolysis. So the most severe is beta hemolysis, where the RBCs are completely light. So we see the transparent halo around the very small colonies of streptococci that don't have tendency usually to fuse together. So, and then the second type of hemolysis, alpha hemolysis. So the RBCs are not lysed completely. We have modification of hemoglobin into hemoglobin. And it has the greenish color, and it gives the greenish color to the blood agar. So it is called viridan streptococci. Can you see here? Viridan. That is quite often alpha hemolytic. So we don't see any transparent zones around it. So then, streptococci may be inflammatory ontogenous streptococci that cause particular diseases, pulpits, and the microfluoride purulent separative inflammation are hemolytic streptococci from the group viridan streptococci. And they give rise to a green discoloration. That's why it is called viridan of the blood agar around the colonies. So, and they also cause acidification of the medium because they have very high fermentative activity to break down sugars. Then they also perform synthesis of polysaccharide dextran that serve as bridges holding together microbial cells on the surface of the tooth to produce plaque. So it turns to caries. Then what are other diseases produced by streptococci? Streptococcus pyogenes. This is a group A beta hemolytic streptococcus may cause a lot of purulent infection. These are pharyngitis, scarlet fever, pyoderma, bacteremia, and also some non-purulent diseases that are rheumatic fever and glomerulonephritis. We discuss a bit later on, okay? Then the second important example is Streptococcus agalactiae from group B according to Landsfield classification. They may cause neonatal infections, urinary tract and wound infections. Neonatal infections are really very severe. There's a very often happens in the labor house and or in care facilities. Then Streptococcus pneumonia. These are very important human pathogens that may cause pneumonia, sinusitis, otitis media, meningitis, and bacteremia in small children. And of course, the group of viridan streptococci that may cause dental caries. So, Streptococcus pyogenes. The thing is that diseases caused by these streptococcus are divided into two broad categories. First, these are separative diseases or separative conditions when pus is formed. In non-separative sequela of previous separative infections. So what separative infections or conditions is caused? For example, the multiplication of it in this throat. It may cause very severe so-called strep throat. Then on the skin, it will cause erysipelas, necrotizing fasciitis, and impetigo that is shown here. And also it may cause systemic diseases as a scarlet fever because except for pharyngitis, this Streptococcus pyogenes is able to produce extra erythrogenic toxin, and toxic shock can be caused by this toxin, and that is really very serious disease. So what is non-separative sequela of the diseases? The thing is that During the repeating streptococcal infection, the antibodies can be produced, but these antibodies can cross-react with a certain host tissues. So these may indirectly damage the host tissues. For example, the antibodies may cross-react with the heart valve tissues. So if these antibodies due to circulation bind with these heart valve tissues, then the systemic inflammatory response caused by macrophages and complement system may damage the cells of the heart, the cells of these tissues. So, and may cause very severe non-separative complications like rheumatic fever, or glomerulonephritis. Some examples of suppurative infections. This is necrotizing fasciitis. So, and regarding the ability to spread rapidly in a body, Streptococcus pyogenes is called group A. Streptococcus is called flesh-eating bacteria because of the tendency to spread and cause dramatic, life-threatening disease. So then this is called erysipelas from erythas, red, and pala skin. This is skin infection marked by the very severe erythema, swelling, bullous formation, fever, pain, and swollen lymph nodes. It is caused again by group A Streptococcus uh, and group C Streptococcus anguinosus and group G Streptococcus anguinosis. Then typical strep throat, streptococcal pharyngitis, usually caused by beta hemolytic streptococci in children from 5 to 15 years old. And you can see the tonsils become tender, develop white pus-filling lesions. But then sometimes, if we have this type of infection with streptococcus, that may produce additional so-called pyrogenic toxin. So then the disease may be turned into so-called scarlet fever. So because the activity of pyrogenic toxin and the dissemination of this toxin in the blood. So the incubation period for this disease is very short, and the temperature can be very high, and the very typical symptoms are strawberry tongue. You can see the strawberry looks like, or the tongue looks like a strawberry, right? And the distinct pink red rash will be developed within 24 hours in a body. So what are the virulence factors of streptococci? So first we take a look at the capsule. Some of streptococci may have very thick capsule, but quite often it can be made of hyaluronic acid that protects from phagocytosis because hyaluronic acid is not recognized as foreign substance, so the antibodies are not produced and don't mark this capsule for phagocytes. So phagocytosis can be not ineffective. Then we have fibrils of teichoic and lipoteichoic acids. You can see here that may serve for colonization. And pay attention that these fibrils of lipoteichoic and teichoic acids may serve as a substrate for M protein. The M protein are fixed on them. And the M protein is very important factor of colonization and also antiphagocytic factor because it inhibits the complement activation. Then the M protein is also considered as rheumatogenic because they contain antigenic epitopes that cross-react with heart muscle uh, antigens. So this is the point when the anti-M antibodies may bind with the heart muscles to cause autoimmune rheumatic carditis or rheumatic fever. So then the M protein also support uh, the adhesion of the streptococci to the host epithelial cells. Then one more type, F proteins, can be incorporated into the cell wall of streptococci. And this is F fibrinonectin binding protein, also mediate the adhesion to epithelial cells through binding to fibrinonectin of the surfaces. And also streptococci are very uh, important members of the biofilm formation on the surface of human tissues or some artificial like catheters. So they are capable of producing so-called exopolysaccharide, the substance that is similar with that of the cell wall of the peptide cell wall or peptidoglycan layer, right? So, and those exopolysaccharides may serve as a core of the biofilm formation on the surface of the skin or artificial materials, right? And that's how you see it may happen. So the matrix of bacteria with the polysaccharide, and then it serves as an anchor to other bacteria to bind and to form the biofilm on the surface of the substrate. And you can see here that is a biofilm formed on the surface of the tooth animal, and see it with the acid production out of fermentation. It causes demineralization, and this is a white spot. See, this is here. So on the tooth animal, and then caries formation. Uh, mm, so a few words about the streptococcal toxin. So the pyrogenic or former erythrogenic exotoxin, streptococcal pyrogenic exotoxin. So uh, it the production of the toxin is a result of lysogenic conversion of the streptococci infected with the bacteriophage that keeps the information about this toxin production. And it may also cause delayed hypersensitivity and shock and the organ failure. That's a very dangerous toxin. We consider three types recognized based on the antigenic structure ABC, and these toxins also may act as superantigens, and they stimulate binding of the macrophages and T cells through non-specific interaction. And if 20% of T cells may be stimulated, so we have cytokine storm and the disbalance, so that may cause tissue and organ failure. So some more of the virulence factors found in streptococci. We may have at least two types of streptolysin, so-called streptolysin S and O. The first one is oxygen S stable, cannot be inactivated by oxygen, and this is leukocidin that can cause lysis of RBCs, leukocytes, platelets. But usually the immune response is not developed against streptolysin S, while the streptolysin or this oxygen labile leukocidin that also can cause lysis of erythrocytes, leukocytes, platelets, but it is immunogenic. So antibodies against streptolysin or so-called SAO antibodies may be used in rapid SASO test that can be performed at the bedside to know whether it is streptococcal infection or not. So that is a very important test for antibodies. Then streptococci may produce streptokinases that cause lysis of the blood clot or fibrinolysin. Then they may produce DNases that they depolymerize cell-free DNA in pus, because pus is very viscous, right? So, and then to release bacteria, to let bacteria to be free from the pus and spread, streptococci may produce these DNases to cut cell-free DNA that make pus more viscous. So, laboratory diagnosis. We have stated that the usually we need to perform culture. And usually, if we are talking about separative infection, we take a specimen from the patient and try to isolate bacteria in the laboratory. In case of non-separative complications of previous streptococcal infections, we may perform uh, tests that allow us to find these antibodies, abnormal antibodies against streptococcal components. So, if we would like to culture streptococci, it is not so simple thing like to culture staphylococci. Streptococcus, especially pyogenes, it has very exacting nutritional requirements, and they won't grow on simple meat there, and we have to use enriched meat there. And quite often we may use even enrichment medium. For example, if you would like to isolate streptococci from the bloodstream, we have to perform blood culture in sugar broth first, liquid culture, to let streptococci first to grow there, and only then we may subculture it on the solid medium. Otherwise, streptococci will not grow very well, and the only enriched medium is used for their growth. Then when we get the colony, so we perform catalase test to be sure that this is streptococcus but not staphylococcus. And then we perform Landsfield identification, so serogrouping to find the specific streptococcal antigens. And also we may perform the the culture on the blood agar to demonstrate type of the hemolysis. Uh, if we are talking about microscopy, is not of high diagnostic value. For example, if we check the throat infection, because streptococci are normally occur in the oral cavity. So, of course, some ELISA test can be used to detect the particular antigens in the throat swabs. But then the rapid test, serological test can be used to demonstrate the presence of antibodies against streptolysine O, right? ASO antibodies. So how a body defend itself from the from streptococci? So Streptococcus pyogenes usually cause exogenous infections following viral diseases or disturbance of the normal flora. So in the normal human, the skin is a very effective barrier. If we have intact skin, this work very well against invasive properties of streptococci. And usually non-specific defense mechanisms that we have on the surface of the skin prevent bacteria from penetrating beyond the superficial epithelium or the upper respiratory tract. But if the skin is damaged, then there's a very easy way for streptococci to cause infections. So the phagocytic system also work very well against streptococcal invasion. Because organism can be opsonized by activation of the classical or alternative complement pathways, and the anti-streptococcal antibodies can be seen in blood. So we mentioned that hyaluronic capsule of Streptococcus pyogenes may allow the organism to evade opsonization, because the antibodies will not produce against the capsule when it is engulfed by macrophages. See, streptococci, nothing happened with it. This is the stages of incomplete phagocytosis, where bacteria are not digested due to the antiphagocytic capsule. So how do we perform treatment? Penicillin is still quite effective in treatment of group A streptococcal disease. It is important to identify and treat group A streptococci to prevent the non-separative sequela that may occur as a result of multiple previous streptococcal infections. Vaccines are not really produced and developed. So some are tested, but still we don't have very good protective vaccines. Because if antibodies are produced, so then it may cross-react with the heart and responsible for rheumatic tissue. So that is the problem with immunity against streptococci. With this, I would like to finish. And please, this is the lecture that you use in the special microbiology, and the only one lecture left that will be delivered on the 13th of June, and that will be the last one. Okay, so my dear, see you next time. Goodbye.